When a sample is named, the expression of genomic DNA or RNA in the technique that allows that study of molecular genetics of an organism and the PCR technique is used. It's important to know the nature of the cell of which will be extracted the interested nucleic acid for to select the correct process to the purification. The PCR technique is the reaction of the polymerase chain in the replication mechanism of the DNA. Because of the polymerase enzyme is used to generate copies of determined sequence of interest through primers or principal application in diagnostics. The PCR consists in three principal steps: the decentralization of the DNA, alignment, and polymerization. The utility for the PCR method is, as we have many samples, it's more easy to identify some bacteria or virus. Also, we can make genetic studies or to identify a person.
There are different types of PCR. For example, nested PCR, that is a modification for PCR intended to reduce non-specific binding sites in the products due to the amplification of unexpected primer binding sites. The RT-PCR are the one that is used inverse transcriptase to do DNA synthesis of RNA complementary. Real-time PCR that allows the quantification of DNA or RNA present in the original sample. The multiplex PCR, that is a simultaneous than one sequence. For these, two or more pairs are combined in a single tube, and together with remaining ingredients, a reaction in sufficient quantities amplified simultaneously. There are others PCR, like hot star PCR, colony PCR, polymerase cyclic assembly, and double digital PCR. How to do the primer? You have the sequence of interest. Then you have to select 18 to 25 nucleotides. Once you are selected nucleotides, you have to count at the percentage of CGs, 60, 80, 40. Then you have to do the reverse complementary for the primer forward. Once you have the reverse complementary, you have to choose a restriction enzymes you have in the plasmid. Once you have the restriction enzymes, you have to insert that in the primers you are done.
The steps we followed for doing the PCR was: first, we needed to defrost the DNA sample and the Taq polymerase kit into room temperature. Then we needed to consider the quantity of solution we're going to use for the mix buffer. For this, we remembered that we were going to use 3 tubes. One that had the DNA sample, another one with a positive control, and the last one with a negative control. Then, after we had our 3 tubes ready, we proceeded to go to the thermal cycler and program it. We program it first with 94 degrees during 30 seconds for the naturalization of DNA, 58 degrees and 30 seconds for the primer alienation, 68 degrees during 30 seconds for the polymerization, and these 3 steps were going to be repeated during 30 cycles. Then we needed to add 68 degrees during 5 minutes, and finally, when all the cycle was completed, the thermal cycler will be at 4 degrees into an indefinite time until we got the samples out from it.
An electrophoresis gel was run to prove that the results of the PCR were the correct ones. So, in the gel, we need to see a band that allows us to determine the sex in humans, because ZFX and ZFY are homologous genes on the non-pseudo-autosomal parts of the sex chromosomes. In this table, we can see the sequence of the primers, forward and reverse, their temperature of melting, the alignment temperature, the target sequence, and the size of the amplifier. The secondary structures that can be formed with the primer forward are a hairpin and a homodimer. The secondary structures that can be formed with the primer reverse are a hairpin and a homodimer. Finally, the heterodimer that can be formed is the next. It's necessary to mention that if the delta Gibbs energy is more negative, it means that the secondary structures are easier to happen. Image 1 shows the electrophoresis gel for the analysis of the PCR. Lane 1 has the Oxygen's 1 kilobase ladder DNA marker. Lane 2 doesn't have any PCR product. In this lane was added 47.3 microliters of mix and 2.7 microliters of DNA. Lane 3 is the negative control. In this lane was added 47.3 microliters of mix and 2.7 microliters of water. And lane 4 is the positive control. Here was added 47.3 microliters of mix, 1.7 microliters of water, and 1 microliters of PCR. It has some bands. The most concentrated has a length of 400 base pairs approximately.
As you can see in the electrophoresis gel, 4 lanes containing PCR products can be identified. There are many reasons why the PCR product fails to distinguish adequately, as a possible contamination of the sample or that the DNTPs were defrosted more than 3 to 5 minutes. To improve the PCR process, changes could be done in the preparation of samples before placing them in electrophoresis gel, as properly calculate the concentrations of the reagents depending on the number of cycles that you want to perform. It must also take into account that if the DNA template is not purified, then it may intervene in the reaction. Also, we could change the condition in which the primers could hybridize with DNA template. However, one of the most important processes is when optimal temperature is lowered so that the polymerase starts its function. In this process, we must be very careful before placing the sample in the thermocycler, because it is necessary to leave the PCR product at 4 degrees for polymerase to not begin their activity ahead of time.
The realization of polymer chain reaction allows us to integrate different techniques used in the laboratory. Since previously from the extraction performed, the template necessary were taken and they are added to the solution prepared enzymes, like DNA polymerase. Nucleotides, primers, and buffers need the reaction. In addition, the preparation of agarose gels was required to run an electrophoresis, thus analyzing the quality of the product.